You have a new job in the pharmaceutical industry, you have a job in academics, and you know that you're supposed to write grants, attract students, but how do you do that? How do you make the changes and different transitions throughout your career? And while you can ask colleagues, having a track where that is the theme of the discussion can be very useful. And what is nice about this is that because it's at Pittcon as opposed to a specialty meeting, the people attending are a little bit broader in terms of their backgrounds.
Hello and welcome to the Pittcon Podcast. In this episode, we'll be speaking to Jonathan Sweedler about professional and career development within the sciences. So thank you for joining us here today. The first question is, please could you introduce yourself and share a bit about your journey to becoming the James R. Eiszner Family Endowed Chair in Chemistry at the University of Illinois?
Thank you. Well, I'm Jonathan Sweedler. As you mentioned, I'm the Eiszner Chair at the University of Illinois Urbana-Champaign in the chemistry department. I'm also this year acting head of the department. I also have appointments in neuroscience, and so I have graduate students that graduate in neuroscience, bioengineering, physiology, biochemistry, and neuroscience. And so there's lots of different programs that I impact with. But I'm trained as an analytical chemist. I did my undergraduate degree at University of California at Davis, and I did actually NMR work with Claude Lamar, and then also at the Livermore Lab with Tomas Hirschfeld, PhD at the University of Arizona with Bonner Denton, and that's actually where I attended my first Pittcon, and then I moved to Illinois, hard to say this, but a third of a century, 33 years ago. Just very briefly, my work is small volume analytical chemistry applied to the brain, which is why I have those different appointments.
Perfect, thank you. And your research in developing new approaches for assaying small volume samples is incredibly interesting. What initially sparked your interest in metabolomics, peptidomics, and mass spectrometry imaging?
So when I was looking, actually as a graduate student, what I wanted to do as a faculty, I looked, what application would keep me going for quite a few decades, and I thought of the brain. And then I really kind of strategically said, what is it that needs to be done? And I really said, I'm going to come up with techniques that allow very small areas to be assayed, eventually single cells. And so looking around, I did looked and found Dick Zare at Stanford, who was doing some wonderfully exquisite laser-induced fluorescence with capillary electrophoresis. And then between us, we came up with the idea of working with Richard Scheller. And so the two of them together sort of introduced to me the topic. And so that's what started my training. I actually, unlike most postdocs, I sat in a lot of courses and learned neuroscience. When I moved to Illinois, I sort of continued in that area, and it slowly got better. So in the beginning, you would measure 5 compounds in a cell or 10. And if you start doing 30 or 40 or 50, suddenly it becomes, instead of small molecule characterization, it's metabolomics. And peptidomics was measuring the peptides. So sort of the omics scale happened as it became more complete. And so what is kind of interesting is we've been doing that for 30 something years. 2023, the editors of Nature picked the 7 technologies to watch, and they picked single cell metabolomics as the technology to watch. And so, 30 years after I sort of got into the field, it's finally the technology to watch. And now I guess it's 2024. So now it's passé. It's 1 year too late. But anyway, this has been the area I've been working on.
Brilliant. And in your extensive career, you've characterized small molecules and peptides across various animal models. Could you discuss a particularly challenging project and how you navigated its complexities?
It's a great question. Partly the most challenging projects never worked. And so, so I'll take a step back. What I really, a lot of people want to say you're studying the brain and they want to know what disease. And I sort of want to know how it works in health. And as a chemist studying the brain, the question is, how do you make it simpler? And so I started looking at things like sea slugs, which have 10,000 neurons, but they can learn what to eat, what not to eat, how to find a mate. So the same types of behaviors maybe you would expect. And so we started looking at animals across the animal kingdom, the metazoan. And we've worked with quite a few. And some of the hardest ones are when the animals are hard to get. We did some work on Decoding gigas. This is giant squid that's found several thousand feet deep, except in La Nina years. And so then what do you do when you can't get them? And so those are hard because the samples aren't always around, and it's a little bit awkward. The thing that I have found actually kind of interesting and gratifying is actually that no matter what animal we work with, from a human sample to a sea slug to a coral, they still use the same molecules. And so in some ways, as we get better on a simpler system, it really applies to working with, say, primates or humans. And so the hardest projects were the ones in the past. And now that things work well, in some ways, a lot of them are a little bit easier.
Yeah, brilliant. Thank you. And when establishing your research group, what were some of the key considerations and challenges you faced in organising and managing a diverse team of students and researchers?
So, you start out small. you have nobody in your group, you grab a few, you end up with 5 graduate students, then it's more, and then some postdocs. And now I've had 76 PhDs graduate from my group. I have 12 in the group. A few have gotten masters, and so it's almost 100 and about 50 postdocs. So what happens is you add complexity. You start out mostly in chemist. A few years later, I added my first neuroscientist. I think in the last year and a half, I've had people graduate with degrees in 5 different departments. And so the complexity comes a little bit later. I think if you started out by having that sort of scientifically a diverse group, it wouldn't work. And then the other thing that happened, which was sort of by luck, I had 2 wonderful postdocs in the group, Stas Rubak and Elena Romanova, who decided to stay with me. And now they're research professors, and they have, it's not a pyramid where they run the group and I work with them, but they have skills that I never have had, from dissections to keeping the instruments running to some of the software aspects. And so now we're sort of a team, and that really has been fun. And so the group grows to handle the complexity that you have.
Yeah, perfect. And could you share a little bit more about your approach to mentoring and fostering a productive, collaborative environment within your group?
Great question, how to mentor. And so, it's one of the things I'll say is that it takes a while, but you eventually come to realize that the most important output of your group is not the 500 papers, but it's the graduate students and postdocs. I have a particular mentoring style where in some ways, I don't like people to tell me what to do. So to the best of my ability, I like to give people the freedom to do what they want. And I have to qualify that. That's also, they have to follow compliance and safety if we're working with animals. So those aspects, you get in a car, you wear a seatbelt, you have to follow the safety rules. If you're working with animals, you have to follow lots of protocols. But I want to give people the option to fail. I want to let people have the option to learn. At the same time, I want to have a supporting environment. One of the keys, I think, is that people specialize within the group, and I want to make sure that they work together. It's not a competitive group. You reward teamwork and success, and so that works well. The other thing I would say is that my group has people that want to go in industry, some that want to go into academics. If they're interested in an academic career, then papers might be important, and maybe they take a little longer and get a few more results. And so it really, I try to adapt to the individuals.
And in a talk that you're giving at Pittcon this year, you mentioned the steady addition of new projects to avoid becoming stale. How do you balance the influx of new ideas with the ongoing projects in your lab?
It's a great question. And I think everybody has problems with this, and so do I. You add projects, and that happens all the time. I didn't start out doing a lot of mass spectrometry, but capillary electrophoresis with laser-induced fluorescence detection. You add more projects, and you have to get rid of some. Some happen naturally because they're no longer funded, but there have been, half a dozen times where I have a successful project, people working on it, and I make the decision that as the people graduate, as the funding ends, I'm going to end that. And that's just simply because of time. I was working with capillary scale NMR, and you have to go to separate meetings. Maybe you could go to Pittcon, but you go to the experimental NMR conference instead of the mass spec meetings. And to stay up in another field is a lot of time. And so in some ways you say, okay, this project has gotten as far as I can. It's been 8 years or something. And so now it's time to change. And so you be deliberate, and it's hard because nobody wants to stop something. You see other people move into the field, but that's the only way to keep, as far as I can tell, the only way to keep on adding projects.
And you've mentioned the importance of scientific papers. With over 100 manuscripts published, what do you believe is the key to producing high quality, impactful research consistently?
Obviously, to publish high impact research means that you spend a lot of time thinking about the story you're going to tell, the project, even before you assign it to graduate students. I sort of get the feeling that some people look at what they have and say, what story do I tell? But in some ways, I want to make sure that whether a project is completely successful or not, a graduate student can publish it. And that's part of my responsibility. And so I think about the project all the time. Obviously, the key then is good project ideas, obviously enough money to get it done, and fantastic graduate students. And if you have those, then you get impactful research.
Yeah, definitely. And within your career, you've taken on further roles, including associate editor, university administrator, and professional society officer. How do you manage your time across these diverse responsibilities whilst maintaining focus on your primary research? So
one key is that all of those weren't done at the same time. And so you can look at a long list. And so, but with that said, I've been an administrator for more than 2 decades, and that takes time. There's a little bit of benefit because it buys out some of my teaching, but you have to make a decision. So I enjoy editing, and so I was an associate editor for analytical chemistry for I think, about 8 years, and then I became the editor 12 years ago. And so that's a choice you have to make. And it takes a lot of time. I figure that's about 18, 20 hours a week. And I do that, it's every week. And so you have to decide if it's rewarding. I really enjoy that. And the interesting part about that is, for example, 2023, Analytical Chemistry, the journal, ended up with the most papers ever submitted, and January is the busiest month we've ever had. That's good for the journal, but it also is a little daunting when suddenly there's a 5 or 10% increase in submissions, and it's like, wait, where did that time come from? So even within the same job, the amount of time sort of varies, and if things are going well, it may increase. So you're always having to evaluate how you're spending your time.
Was there any particular reason for that increase, or was it just...
So the number of submissions, it's a good question. It goes up a little bit because it is for all journals, but in the last part of 2023 and into 2024, when I look at other ACS journals and others, they haven't seen that increase. So I'm not actually sure. Is it, it seems to be continuing. So sometimes there's a sudden increase after an impact factor increased. You suddenly go from 7.2 to 8 and people submit, but that's not what happened. So I can't tell you the increase. The reason is a little interesting.
Pittcon Perspectives, unforgettable Pittcon moments from the experts.
Obviously, we're here at Pittcon 2024 in San Diego, and you've mentioned that you've attended many Pittcons over the years. Over the years, has attending Pittcon influenced your research directions or collaborations?
So Pittcon, I actually started as a graduate student. I think it's hard to believe, but like in 1984, I went to my first Pittcon. So if I'm, 40 years ago. So I go to meetings like ASMS, and it's mass spectrometry, or I go to the HPLC meeting, and they're fairly narrow focused, and you learn a lot about that focus. Pittcon, sort of just like being a journal editor, makes you see things outside of your own field. I see people I know in the field of analytical chemistry, but don't go to the meetings I do. And so that's absolutely fantastic. Pittcon gives many of the awards. It's become the award meeting across many different societies, and that's always fun to see the best and the brightest and the young people succeeding. So going to award sessions is wonderful. And then there's a lot of national committees or international committees. They might meet here or at BCEIA or maybe urinalysis, but in the U.S., this is a very good meeting to bring those together. And so there's a lot of reasons for going, but I really like the diversity.
So this year we've got a new scientific track of the professional and career development track. Why is it so important for a conference like Pittcon to highlight professional and career development? And what do you hope attendees can learn from your talk as well as the others on this track?
So it's a new track. They have done some professional development. I think it's important because there is, in some ways you become you have a new job in the pharmaceutical industry, you have a job in academics, and you know that you're supposed to write grants, attract students, but how do you do that? How do you make the changes and different transitions throughout your career? And while you can ask colleagues, having a track where that is the theme of the discussion can be very useful. And what is nice about this is that because it's at Pittcon as opposed to a specialty meeting, the people attending are a little bit broader in terms of their backgrounds. So the session that I'm involved with has people that have started companies, that are working in industry. I'm talking about an academic. And so hopefully the diverse perspectives means that you can pick up some advice that actually may help you a little bit more than if you were very narrowly focused.
What makes Pittcon so special, and why do you return year on year?
So I've probably gone to Pittcon more than any other conference in my career. And so it is true that I return many times. It's the diversity of topics. It's the center of the field of analytical chemistry, and it really does bring people together across the entire field, and I think that's critical. And so, why Pittcon? You might ask, how did that happen from the 1950s when it's, 75 years ago? But in the last 25 years or so, this has been the conference to bring the field together, and I think that's one of the important reasons for attending.
Yeah, and this year is the first time that Pittcon is on the West Coast. What have you been most looking forward to about Pittcon in San Diego?
Well, that's easy in the sense that I live in the center of Illinois, and normally the temperatures are pretty cold. We just had snow last week, and so coming to San Diego is always a treat. And this time of year, I can tell you, Pittcon has been in Chicago, which is a wonderful city, but really Chicago in February, March. And so, yes, if we're going to have a meeting, it's nice to be able to walk around outside and not wear a coat.
Yeah, definitely. And as we mark the 75th anniversary of Pittcon, could you share your first memory or experience of attending this conference and how it impacted your view of the scientific community?
Boy, so again, because it's 40 years ago, I attended a few times as a graduate student. I was over, it was my first, one of my first places I ever gave a talk. And so being able to give a talk and see all the luminaries in the field present was just an amazing experience. Getting to go to a few receptions and suddenly being able to talk to people that I've been reading their publications was a lot of fun. And people are very approachable. And so that, those are the reasons that really, stick with me. I do remember the first Pittcon, there was a reception for the Analytical Chemistry Journal. And I went in and, met the editor at the time. And I certainly, I certainly had no idea that, 30 to 40 years later, I would be the editor. And so it's just been a fun full circle as I've attended Pittcon.